Hello everyone. Today in this basics of genetic part 4 we are going to discuss, we will carry on the class right from a part 3 where we had stopped. So what all things we are going to study today for today's lecture. So first of all we are going to speak about linkage. Then we are going to speak about linked genes. Then third part we are going to speak about linkage group. Then we are going to speak about types of linkage. Then we will start with crosses. Means we will start with Mendelian, we will speak about Mendel and its crosses, all the practical, all the experiment which were done by the Mendel. So we will speak about monohybrid, dihybrid and all the crosses. So we will just start with the cross today and then we will continue in the further upcoming classes. So today let's begin about linkage. So in the previous lecture we spoke about actually what is linkage. So if we take a chromosome and if I mark two genes on chromosome, let us say that this is gene A and gene B. So you can see that A and B, these two genes are very close to each other. If I say they are very close to each other, I mean to say that they are linked to each other. They are linked to each other. So these are linked genes. These are linked genes. And the distance between these two genes is very less. So distance is very less. Now one thing I will say, linked means as much as closer the two genes are. If they are very close to each other, then we will say that they can inherit together. So these two genes here, they are very close to each other. Very close to each other. If they are very close to each other, means if they are very close to each other, means they can inherit together. Okay, they can inherit together. So this is the concept of linkage, or you can say linked genes. Okay, so there can be many genes, not only two, there can be several genes which are linked together. If they are linked together, so they have a capacity, or we can say tendency to inherit together. Okay, they have a tendency to inherit together. So if they are very close to each other, then they will not participate in crossing over. So they won't participate in crossing over because it will be very difficult in this lesser space to make chiasmata. Okay, so it's very difficult in this smaller space to make chiasmata. Even I told that if there are two chromosomes, one is paternal and the maternal chromosome, homologous chromosome, that is, we can say, so their chromosome part get exchanged. Okay, if their chromosomal part get exchanged, means there is crossing over. So for an exchange of chromosomal part, the genes must be for crossing over, for variation, the genes must be little bit far away. If there is this chromosome and let us say that this is A gene, and you can say E gene and F gene. Okay, I will change this. Suppose this is the chromosome and this is E and this will be F. So you can see the distance between these two genes is higher. If the distance is higher, means the genes are very far from each other. That means they are not linked. The bond is not stronger. Here the genes bond is weaker bond, but here the bond is stronger bond. What I will say here, it is strongly bonded. So it is strongly bonded here, but here it is weakly bonded. Okay, so if they are weakly bonded, then here the crossing over will be maximum. The rate of crossing over will maximum, or you can say the percentage of crossing over will be more. They are weakly bonded. So here the crossing over will be higher, and here it will be least or it won't occur at all. This side. So these are the two concepts. See, you can say this is a part A and this is a part B condition. So both the conditions are different here, part A and part B. Okay, so I will formulate a simple formula for you. Okay, if the linkage is more, then crossing over will be least. So I am giving you one proportional. So linkage is inversely related to crossing over. Okay, and speaking about the distance, so if the linkage is more, see here the linkage is higher. Here if the linkage is higher, means the distance between two genes is lesser. So again we will say that if linkage, if there is a need of linkage to be higher, so the distance between the genes must be lower. So it is again inversely related to distance also. Okay, and it is also inversely related to recombination. Now crossing over, or you can say recombination, one and the same thing. So this is the short formula for you. Now I will show you how this is true. See, if I take one chromosome, okay, and if then I take one like this, this A gene, E gene and F gene. Now if you can see, these two genes are very far from each other. Okay, they are very far from each other. So far, okay, I will write far apart, like they're very far. So if they are far, means what? Linkage is less. Means linkage is less. If the linkage is less, means crossing over will be higher. Okay, and I told far means what? Distance is higher. Distance is higher, or you can say they are very far apart from each other. Okay, and if the recombination, like if you can say crossing over is occurring, means there will be recombination. So recombination will also be higher. And if I take if I talk about other two genes, suppose here there is A and B. Now you can see the distance is lesser. I will write D for distance. Okay, if D distance is less, means L, that is linkage. Okay, I am marking the short forms. Okay, if the distance is less, so the linkage will be higher. Okay, so the linkage will be higher, then the crossing over will be less, and R means recombination also will be less. So here there are two scenarios for the same thing. Okay, for the linkage topic. So this is about linkage and the linked genes. Now further moving towards the type of linkage. Basically, like the concept of linkage was brought by the scientists like the Betson, Punnett and T.H. Morgan. Okay, these people discovered linkage. So the two people are from this, like we can say two people are from botany and one is from zoology. So the Betson and Punnett, these two are like from the botany field, and the one which is T.H. Morgan, who actually worked on the Drosophila, he is from the field of zoology. Okay, so further we will speak about the next part. So you can say the types of linkage. Types of linkage. So basically there are two types of linkage. One is complete linkage, and the second one is the incomplete linkage. So what happens in complete linkage, I have already taught, but I will actually tell you now here what I have taught. This is the concept of complete and incomplete, but let's take it here. Okay, so in this topic of complete linkage, what happens is that the two genes are very close to each other. Okay, so if I take this chromosome, so the two genes are very close to each other, gene A and gene B. They are very close to each other. Since the genes are very closely related, okay, closely related genes, if the genes are very closely related, then the crossing over will be less. As now if I say close related means what? The linkage is higher. Linkage is higher. If they are very close related means the bond between these two genes is stronger. They are strongly related also, we can say. So they are very strongly linked to each other. Strongly linked. If they are very strongly linked, then there will be no possibility, we can say lesser possibility to no possibility of crossing over. If crossing over is not there, then there will be no recombination. This is about complete linkage. We can say one example as in X chromosome of Drosophila male, we can see this example of complete linkage there in Drosophila male X chromosome. Speaking about incomplete linkage, so in incomplete linkage, what actually happens is either the two genes are very far from each other. This is the first thing, like they are very distantly located, means they are very far from each other. So if I take here gene E and here gene F, means they are very far from each other. Or we can say that the two genes are on different, like one gene is here A and one gene is B, like here they are far apart as this way, we can say. But in the syllabus they are only speaking about this, only in one chromosome they are speaking about, but the chances can be that the two genes are on different chromatids. Okay, or they will be E and F will be very far from each other. So the distance between these two genes will be very far, longly, we can say far apart genes. Okay, and then we can say if they are very far from each other means chiasmata can be formed, and hence crossing over will be higher. If crossing over is occurring, then recombination is also higher. Okay, and if they are very far from each other, like they are very far, so linkage will be less. Linkage will be less. if they are very far, see if two things are very far, so they are weakly related. Now suppose if your friend is in some different area, suppose the friend is in other country and you are in other country, means you are very weakly related to each other now. So the bond is not that strong, we can say. So this is weakly related, weakly linked genes, we can say rather. Weakly linked genes. Okay, so this is the two difference between the incomplete and complete linkage. These are the two topics about it. So I hope the types of linkages will be clear. This is two topics which you need to know: complete linkage and incomplete linkage. Now the linkage can also be on the sex chromosome. If the linkage is on the sex chromosome, then we say it as sex linkage, as the linkage is present on sex chromosomes. Okay, now if I speak about X and Y chromosomes in humans, so see, this is, say, X chromosome and there will be a Y chromosome. Okay, and if I say that, see, these two parts, this of X and this of Y, so this is X and this is Y, so these two parts are homologous to each other, this and this. These two parts of X and Y chromosome, they are exactly same, so we say these two parts as homologous chromosome. Okay, this and this part, we say it as homologous chromosomes. And the above two parts, okay, instead of saying chromosome, I can say homologous regions. Homologous regions. And this and these two parts, these are not homologous, so we will say non-homologous chromosome. As you can see that they are not structurally same, basically. Okay, morphology wise they are not same, so we will say non-homologous regions. Okay, now on the non-homologous chromosome and on the homologous chromosome, there are presence of two different types of genes. Okay, so on like this region which I am telling, like non-homologous part, there is a presence of complete linked genes, or we can say complete sex linked genes. Okay, complete sex linked genes. If there is presence of complete sex linked genes, then the crossing over will be less, as on this non-homologous chromosome region there is complete sex linked genes. So what will happen here, the crossing over will be less, or you can say there will be no crossing over. Okay, means now I told you what is complete linkage, means the genes are very close to each other and all. But if you can see, I had also taught one thing, that whenever there will be homologous chromosome, these chromosomes are only responsible for crossing over. Means the non-homologous chromosomes, they will not participate in crossing over. Okay, जो भी like whoever are homologous chromosomes, they will participate in crossing over, and the chromosomes which are non-homologous to each other, they will not participate in crossing over. Okay, non-homologous won't participate. So if we talk about the X and Y chromosomes, there we are seeing two different regions. Okay, here we are seeing two different regions. One is non-homologous region and other is homologous region. So if we speak about the homologous region, so these regions will take part in crossing over. Okay, and as I told that on non-homologous region there is a complete sex linked genes present. Here in homologous region there is a presence of incomplete linked genes. Which genes are present? The genes which are present on this homologous chromosomes of X and Y regions, these genes are known as incomplete sex linked genes. Okay, incomplete sex linked genes. Okay, so these are incompletely linked. If the genes are incompletely linked, they are very far from each other, they are undergoing crossing over, they are undergoing recombination, and that's what we are speaking here the same. Just we are saying that the genes which are present on the sex chromosome, those genes, if they are linked together, then they are known as sex linked genes. Okay, so if they are very far from apart, like they are very far from each other, the two genes are very far, so we will say incompletely sex linked genes which are present on sex chromosome. So we are saying sex linked genes. Okay, so these are on sex chromosomes, so we will say it as sex linked genes. They are linked to each other, and that also the distance between them will be very far. They will be very far from each other. If they are very far from each other, that means they will not inherit together. Even here also you can write that this is like they are very close to each other, these two genes. I will highlight the okay, wait 1 second. So I will draw it again once again here. See, so what I was saying here, okay, see here, so these two genes are very close, so we can also say that they will inherit together. I will mention here a point that these genes will inherit together. I will write this. Genes will inherit together as they are very close. But here if they are very far, so we can say if they are very far, then genes won't inherit together. Genes won't inherit together. Okay, same thing you can say here also, that if the genes are incompletely linked, that means they won't inherit together. And if they are completely linked, means they will inherit together. Okay, they will inherit together. Understood? So this is about the complete sex linked and incomplete sex linked. Okay, I told that all things. So this is the first part. Okay, so we spoke about linkage, we spoke about linked genes, we spoke about the types of linkage. Now we will speak about this, that is linkage group. Now what is linkage group? Now linkage group means if there are linked genes, okay, if there are linked genes, so these linked genes will inherit as a group. Okay, if there are some genes, we can say specifically they are linked genes, so these genes will inherit together as a group. Okay, if suppose there is A, B, C, these three genes are very close to each other, so these three genes will inherit as a group. So such group which will inherit together in a cell during the during the time of cell division and all process, so they will act as a whole group and they will inherit. They won't be inheriting independently, means it won't happen that this A is moving alone, or this B is moving alone, or this C is moving alone. No, this is not going to happen. Okay, this is not going to happen. What will happen, these three genes will inherit as a group. Okay, so they will inherit as a group. So such group which will inherit together, that is known as linkage group. Okay, that is known as linkage group. Okay, so this is about linkage group. Now suppose if somebody asks you what is the number of linkage group present in humans, so I will tell you this formula. So number of linkage group is always the haploid number of chromosome in that organism. Okay, is always haploid number of chromosomes, in that organism. Number of linkage group is equal to haploid number of chromosome in that organism. Now suppose in humans there is 46 chromosomes as a whole, and we say that there are 23 pairs of chromosomes. So the haploid number of this will be 23. So we will say that in humans there is 23 numbers of what? Of linkage group. 23 number of linkage group. Okay, so it is always the haploid number of that particular organism. Now if suppose we speak about the garden pea plant, okay, so in case of garden pea plant, what actually happens is that they are having 14 chromosomes. 14 chromosome means what? 7 pairs of chromosome. Okay, 14 chromosomes are present in what we are speaking? We are speaking about garden pea. That this is the pea which was used by Mendel, okay, for his experiment in genetics field. Okay, so this is 17 chromosomes, then we will say haploid is 7, so there is the presence of 7 number of linkage group in garden pea. Okay, so this is the entire concept about linkage and linked genes, who coined the word and all. I told everything in this. So the word was like this whole concept of linkage was given by Betson, Punnett and T.H. Morgan. Okay, so and the word I think the word was coined by the T.H. Morgan only. Yes, T.H. Morgan was the person who coined the word also, and even he did the experiment on Drosophila. Okay, and he found about the some variation in the dihybrid ratio. We will speak about someday Morgan's experiment also, but for now we are going to like end the linkage part here, and we will start about Mendel's genetic. Okay, so today we will speak about some concepts in the field of genetics. That is mainly we will speak about crossing. Okay, we will speak about crossing. So there are many crosses. We will move one by one. Today we will speak about only cross, that is monohybrid cross. Okay, so some basics are already taught, and as in forward we will speak about other basics also. So let's first speak about what is phenotype, because you should know these all concepts. What is phenotype, and we will speak about what is genotype. Okay, and I think these two are enough, and we will speak about the dominant, then what is recessive. Okay, and then we will also speak about like a gene also, and alleles, and then finally we will speak about what is homozygous and heterozygous. Homozygous and heterozygous. Okay, so let's begin. So this was about that homologous chromosome, where what about like about this I was speaking. I will take another slide and I will explain about this. So first we will speak about that what is a gene. Okay, we will first speak about that what is actually a gene. So if like we speak about a DNA, okay, so DNA, deoxyribonucleic acid, so this is the genetic material, and if I draw a DNA, a rough diagram of DNA, so if like suppose this part of DNA is giving me some, like it is coding for some protein. Okay, what it is coding for some required protein, for protein. So this is this functional part of the DNA, okay, which is going to express itself. Okay, the expression of a cell is nothing but a protein. So this part of DNA, which is going to express itself. When it will express itself? During the inheritance. Okay, during the inheritance it will express itself. This particular segment of DNA, or we can say a part of DNA which will express itself, that is known as a gene. Now suppose if I have a gene, like if I'm speaking about a gene of height, if I am speaking about a gene known as height, so there will be like it will express as either tall height or it will express as a dwarf height. So there will be a possibility of tallness in the organism, or there will be a possibility of dwarfness in an organism. Okay, so there will be two possibilities of this particular gene. So these two possibilities, okay, these two possibilities of a gene, we can say alternative forms of a gene, it is known as alleles. What we will say this to as alleles. Alternative forms of a gene, that is nothing but known as alleles. What we will say it as? Alleles. Okay, so this is alleles. As here there are two alleles for height, one is tall and one is dwarf. So this is first allele and this is second allele. So there are two alleles for one thing, for one gene. So if I say there is a chromosome, so there will be like two alleles which will be occupying this, like, okay, so if I say that this is gene number A and there will be a suppose gene number B, so each gene will have two or more forms of itself. Okay, if the gene is present here, if the A gene is located here, means their alleles will also have same location. Wherever the gene is located, that location we say it as loci. The position, okay, the position where the gene is present, we say it as loci. Position of gene, we say it as loci, or we can say location. And these alleles will occupy same loci, or you can say identical loci. What we can say? Identical loci means it will be present on the same loci, as you can speak about A and B gene in this particular example. So this is the concept about gene and a concept about alleles. Okay, and the concept about alleles. So now we will move forward towards the another concept, that is, we spoke about alleles. Okay, oh, by mistakenly I cut it off. We spoke about the alleles and we also spoke about the genes. Now let's speak about what is genotype and what is phenotype. Okay, these are all you can say the terminology in genetics. What it is? These all are terminology in genetics. You can write it here that it's a terminology in genetics. Okay. So these are terminologies in genetics. Terminology in genetics. This is very important topic to be known. Okay, so the next part we will speak about what is genotype. So let's speak about the height itself. Now the height was having two alleles known as tallness, or we can say tall, and one is dwarf, or we can also say dwarfness. So this tall, okay, this tall is like, when I say just tall, so it shows that we are speaking about the external appearance of something. Okay, but if I say that this tall is written as capital T means what? This capital T denotes the genes of this, like of this tallness. It is expressing. Okay, so this is the way we write tallness in genetics. Okay, so we can say that capital T, or we can just say that this capital T is the allele for tallness. Okay, what it is? It is allele for tallness. Okay, if I want to denote this dwarfness, so we will denote this as to small t. This is noted as capital T and this is denoted as small t. Okay, so this is about genotypic, means how it looks in at the gene level, whether it is capital T, it can be, now tallness can be capital T, or it can be capital T, small t. Now what is this? I will speak in some time. Okay, and this dwarfness possibility is just small t. Okay, so this is the way, like this is what we are writing, capital T, small t. This is at gene level, it is genotype. Okay, this is known as genotype, means at genetic level how it is, means what type of makeup it is having. Okay, we can say genetic makeup, in short, genetic makeup. So what genetic makeup it is having at gene level, that is known as genotype. Capital T for tallness, or it can also be capital T, small t for tallness. So this is genetic makeup. Okay, so this is genetic makeup. But what if I say that there is a person who is very tall? Means I am expressing its morphology, that how is the external appearance of that person. What I am expressing, I am saying that one person is very tall, means what I am saying that the external appearance of person is tall. So this appearance is known as phenotype. What it is known as? Phenotype. Okay, I can say that the person is tall, or the person is dwarf, or the person is fat and all. Although, okay, we can say, well, however it looks as, means the things which you can observe and say. Okay, if I say that the person is looking like this, means what I had observed it. Whatever things which I can observe and say about someone, that is known as phenotype. And the things which are genetic makeup of something, okay, what it is a genetic makeup, then we will say it as genotype. So the external appearance of a person, whether the person is tall, dwarf, or if I speak about a flower, whether a flower is we can say purple color, white color, or pink color, red color, that is what we spoke about external thing, which the things which are observable. But if at gene level, the genetic makeup of that organism, how it is at gene level, what it is made up of, or you can say what are the constituents, so that is known as genotype. That is known as genotype. I hope that these two terms, genotype and phenotype, will be clear. Okay, now I told that there are two things which are, one is dominant. Okay, like in a house, someone is dominant and someone won't rule the house. Okay, the person which don't rule the house, that person is recessive, and the person which rules the house, that is dominant. Okay, in the same way, in our gene, what happens that if there are two genes, suppose there is capital T and small t, since this is expressing for tallness, which is a favorable quality, or we can say a favorable trait, then this is dominant. Now generally, like we say that tallness is something which is favorable in the nature, and so we say dominant. It doesn't mean that if it is favorable then only dominant. No, this is something which is like, now see, if there are two friends, suppose this is friend number 1 and there is friend number 2. This friend is speaking so much that this friend is not allowing the second friend to speak. means the F1 friend is expressing itself a lot and F2 friend is not expressing itself. So the person who expresses a lot or who has higher expression when it is present with the second person, then that person is dominant, and the person which is getting suppressed, what it is getting suppressed by the first person, okay, then that is known as recessive. Now if there is capital T small t condition, in this condition what is happening, this capital T is expressing itself when it is present with the small t. Okay, what is happening, this capital T is expressing itself when it is present with the small t. Okay, then this small t isn't able to express when it is with capital T. That means this is, the capital T is dominant and the small t is recessive. When you are not able to express when you are with someone, then you are having a you have been suppressed and you are like recessive gene. Okay, you can say recessive allele. And this, when somebody is going on speaking, then that condition is dominant and this is recessive condition. Okay, so this is about two things which I wanted to explain. One is dominant and recessive allele. Hope this is clear. Okay, dominant allele is the allele which is always expressing itself. Okay, I will write it here. Always expresses itself. Okay, and recessive allele is the allele which is not expressing itself. And we can say that dominant allele is masking, what we can say, masking the effect of other gene, or you can say it is masking the effect of other gene, that is recessive gene. Whenever there will be a dominant gene, it will not allow the other gene to express, other recessive gene to express. It will mask the effect of other gene, or you can say other allele. Okay, so this is capital T is one allele and small t is one allele. So this capital T will mask the effect of small t. What it has done? Masked the effect of small t. Means this will get suppressed. If it is getting suppressed, then it is known as recessive. What it is known as? Recessive gene. Its effect is getting suppressed, then this is known as recessive allele. And if this is expressing itself even in the presence of this other gene, like you can say other gene, then this is dominating. Okay, this is dominant gene. So here we spoke about the four concepts. Okay, one was allele, then we spoke about this recessive, dominant. Okay, we spoke about recessive, dominant, we spoke about phenotype, genotype, and now we will speak about what is homozygous and heterozygous condition. Okay, and then we will stop for today's class. Okay, now what is hetero? Hetero means that, oh, it got erased. Hetero means something which is different. Okay, hetero means different, and if I say homo means similar. Homo means similar, or we can say same. Okay, if suppose there is a presence of capital T means two alleles are similar, or we can say there is a presence of two same alleles. Then in such condition we say that it is homozygous condition. Homozygous condition. If suppose there is a presence of small t, that is same, like both the alleles are same, that is dwarf alleles, then we can say again homozygous condition. But what if the allele is present as capital T, small t? If this is the scenario, that is one allele is for which is for tallness, that is dominant, and the second allele, which is small t, that is for dwarfness, which is recessive. If such two alleles are present, then we say it as a heterozygous condition. Heterozygous condition of a gene. So there are two different alleles present here. We can say here two different alleles. Okay, two different alleles, and we can say at this site two same alleles present. Okay, so this is all about the terminologies in genetics. This is heterozygous and homozygous. Now we can also say that this condition, okay, I will take it on, okay, let us speak here only. This capital T condition, we can say dominant homozygous. What we can say? Dominant homozygous. And if this is small t, then we can say it as recessive homozygous. Although it's understood that recessive means the recessive allele will only express itself when two recessive alleles are present, because if the recessive allele is present with the dominant allele, then it won't get expressed. Okay, so it's understood that this is a recessive homozygous condition. Okay, when there is capital T, small t, then capital T will express. When there is small t means these two alleles are for dwarfness. That means the dwarfness, or you can say dwarf height, will be expressed as a phenotype. Okay, so this is about recessive homozygous. This is about dominant homozygous, and this one of capital T, small t, this we will say it as dominant heterozygous, or we can say heterozygous dominant. Okay, you can say dominant heterozygous. Okay, this is dominant heterozygous. So this is all about today's class, and I think from start till now all things are understandable. If you have any doubt about the part 4, you can tell me in the comments. So today we have started with the terminologies in genetics, and we are done with these all terminologies. In the upcoming lecture I will start with the crosses. So for today I will end the class, and thank you for watching this class.